our last 2 hours, we got into the olfactory bulbs. So just as a reminder, we were speaking about olfactory receptors in the epithelium, sending their axons to the olfactory bulb, and started to look at the next brain station, which is the main olfactory bulb, the start of the central nervous system. This is yet another scheme of what we talked about. On the left-hand side, the olfactory receptors expressing the different ORs as represented by the little baby structures here. Different ORs, all neurons expressing the same OR converge onto the glomeruli. And we also talked about the mitral tufted cells, which are the next neurons in this station, which actually are the projection neurons from the olfactory bulb. So from the olfactory bulb, there are actually two types of projection neurons. We will not dwell upon them, but the main ones are these mitral cells. What is unique about these cells, one thing which is nice about these cells, is that they send a single dendrite into a single glomerulus. They also have lateral dendrites. Which means that they can transfer information to other channels of information. Okay, now it's a little bit, it's on the side now. We used to look at this on the other side. So this is the input to the system, this is the output to the system. But one important thing that you should know is that there are many local interneurons in the olfactory bulb which contribute possibly to this input-output function. Okay? Yes.
The bulb itself, when we look and we see the olfactory bulb, we actually see the cell bodies of the mitral cell. What do we see? The glomeruli? Do we see everything together?
Yeah, I showed you last time a nice picture of this. Here, this is, for example, the axons in green coming from the epithelium, converging on these structures. There are no cell bodies here at all. And what you see here are the mitral cells. They send a single dendrite into the glomerulus. What I'm telling you is that now, first of all, there's a single main dendrite going here. There are also lateral dendrites in the olfactory bulb, which means that they can interact with each other, at least in terms of the connectivity. But also there are many, many local interneurons in the bulb. We'll talk about that.
And everything together is the bulb.
Yes. Okay, and these neurons send axons to the next brain station. What we want to know as neuroscientists is, what is the computations carried out in each brain station. And things get very complicated very fast, especially in the olfactory system, okay?
But the interneurons and the lateral connections are only starting at the level of the mitral cells, or it's already happening?
No, so you will see already in the paper that will be presented by Toby, I think, that already there are local interneurons that synapse the axons, the presynaptic axons. Yes, the axons of the receptors in the olfactory bulb, not in the epithelium. So regulation starts very early. Great. So there are interneurons, local interneurons, and there are also top-down axons. Yes.
One microcell per glomerulus?
Excuse me?
One microcell per...
No, not one microcell per glomerulus. The number varies. I would say the numbers are between 10 and 20 microcells per glomerulus. Some probably receive more, some probably receive less, but each microcell sends a single dendrite into the main glomerulus. Okay? So you'll have 10 of these sending axons in here. And there are about 15,000 axons coming in. So there are different levels of connectivity that can impact the computations going on here. One is the local interneurons. These are normally divided to two levels. The level of the input around the glomeruli and the level of the output, which is down here somewhere. So maybe synapses on the lateral dendrites or on the cell body, or maybe axon in the sheath segment, etc. Okay? And these are considered to be, they're called periglomerular because these neurons are around the glomeruli, and granule cells, which are very small neurons. They're very unique cells in the nervous system because they have no axon, and actually their presynaptic component and the postsynaptic component is on the dendrite. So these are special synapses in the brain called dendrodendritic synapses. Okay? So these baby cells, they're very small, have maybe 200, 300 synapses, but they make dendrodendritic synapses with the microcell. And it is thought that there are two layers of inhibition. One at the periglomerula, around the glomeruli, and other one at the granule cell level. However, life is more complicated, and there are many different types of periglomerular neurons, and there are also top-down connections coming from higher or different brain areas. Okay, so you can find axons from all the neuromodulatory systems, acetylcholine, serotonin, noradrenaline, etc. They converge very strongly onto the back to the olfactory bulb, and also feedback connections from the cortex, from the next station. So that said, people have started to look at the basic transformations, the input-output transformations from the olfactory bulb. One of the first papers in the field was a cell paper by our friend Richard Axel in the flies. And what they did is that they made genetically transgenic flies that have calcium sensors in the sensory neuron, and they also made different flies with calcium sensors in their projection neuron. So they could do what I showed you last time that Rachel Wilson did with electrodes, pre- and postsynaptic cells, but this is the start of the story, it's a few years earlier. And because in Drosophila you can identify each glomerulus by name, there are only 60, and you can identify them, this is DM1, this is the whatever, I don't know, DL5. You can test whether the input from the sensory neurons is correlated nicely with the output. So for example, when you give capoic acid, which is an odor, you will get activity of this and this glomerulus. When you give isoamine acetate, which is another odor, you will get a combination of other glomeruli. Okay, this is the combinatorial process. So they did this, and they also did this in other flies, and they did the same experiment, giving capoic acid, and they saw that this is the dendritic signals from the mitral cells, and those of them are called projection neurons. They get exactly the same pattern. Okay? So what they claim is that the olfactory bulb is a simply labeled lines of information. Nothing interesting is going on in between. Whatever comes in, comes out the same way. Okay, so this is just a source of converging information from the epithelium, and there is no real processing. It's a faithful transmission between the olfactory receptor neurons and the mitral cells of the projection neurons in this case. Okay, this was 2003, a big cell paper talking about what kind of transformation are going on. Still to date, you will be amazed that the genetic people who study the olfactory system, they don't care so much about the processing. As far as they're concerned, the main olfactory bulb is like a fly, everything is labeled lines, but this, I would argue, is not the case. Okay? The first lines of evidence that was challenging this is Rachel Wilson that could do electrophysiology. Okay? Because people say, excuse me? Yeah.
What's with the autograph of the gastric effect?
This one?
Yes.
This is just odors, which glomeruli, and olfactory receptors and mitral cells. Okay, so that they just see that there's perfect correlation, more or less. It's not perfect, but almost. Yeah.
It doesn't mean that the mitral cells don't do anything. It's saying they get the same input as the mitral cells.
No, what you see in the calcium signal is the output of the mitral cells, the spiking output of the mitral cells. Yes. What one would expect, if something interesting would happen, of course, is that let's say you get an input from these two glomeruli, but you get an output also from this glomeruli.
But the output is not in the glomeruli.
It's not in the glomeruli, but they have the dendrites. What you see is the dendrites of the mitral cells. So it's a reflection of the glomeruli.
Yes, but it's not the output of the mitral cells. It's the input of the mitral cells. No?
So
They do the
exactly.
computation.
So one could argue it's a mishmash of the input and the output. It's also an output because it's low resolution in time. It also captures the back propagating action potential, things like that. But that was the first, the 2003 papers told us it looks like input, it's a faithful transmission, nothing very interesting is going on. Rachel Wilson, in her science paper in 2005, which was a breakthrough in neuroscience because she was the first one to actually do electrophysiology in Drosophila, found something completely different. Before she did this one-to-one, okay, she just patched olfactory receptor neurons, many, and did a screen of 35 odours. And what she did is she just sorted the responses of the olfactory receptor neurons by the maximum response to the maximum odor. And what she finds is receptor fields which look like this. This is the most significant response of this neuron to odor number, whatever, 17. She always sorted them out by the maximum in the middle and then going to the side by the next one were activated. And she could see these kinds of responses. This is low intensity, or I don't know. So this is the initial phase of the response, later on in time. Okay? Generally, you can see if you look at the very early onset of the response, the first 200 milliseconds, this neuron responds nicely only to one odor and out of 35. So this is quite sparse, where the receptor fields are quite sharp. If you look at the projection neurons of the microbe cells, what she claims is that you get much more broader receptor fields. Okay? So what she claims is that there is not a faithful transmission, but in fact, what can you see here in terms of the connectivity or the effect? Widening of the receptor fields, which probably means lateral excitation, right? So a glomerulus will come in and will excite projection neurons in the cell. So the 2005 in Drosophila, we're still in flies, okay, argues for lateral excitation and widening of the receptor fields at the next station after the olfactory receptor. Yeah.
The plot of the smell of the different odors is organized like that just so it would look like a tuning curve
Yes.
of visualization?
Yes. Okay, and for each olfactory receptors, it's different. You would plot a different olfactory receptor, you will have a different odorant in the middle.
And then she just organized it hierarchy, right?
Exactly. Just by hierarchy, she puts the maximum in the center, the next one to the right, the next one to the left, next one to the right, etc.
It doesn't look like widening. It looks like on the top one maybe, but the two others, it doesn't.
Again, it's not a one-to-one. This is just a pool of all the olfactory receptors that she recorded. She claims if you look here, it's quite peaked responses, not too many odors out of the 34. And if you look at the projection neurons, they're all over the place. So she concluded from this that there is widening of the receptor fields. Yeah. Yeah.
I'm not sure that this really indicates a widening of receptors because in the first instance when you first present the stimulus, right, you still do see a narrow receptive field. It's only afterwards that you start to
Yeah,
right,
but here
right
it's
there.
narrower than this. That's what she claims.
So that's true, but I'm saying you have a narrow response, and then after time, it's only after time that you start to get this widening response. So maybe it's not.
No, her claim is that at any level the ORNs are sharper than the projection neurons. That's the claim. Do you disagree with this claim?
So I agree with that claim, but I think that
So we can move on.
maybe a more interesting thing to look at would be the projection neurons over time, because it seems that there's something happening through the time course there.
Absolutely. Timing is super interesting and super important, but we're not going into this. We're all at the basic steps of what's happening in this brain structure. What computations go on? I'm just giving you where we stand. Okay? This was very hard to do in mice. That said, a group in China, led by Min Lu, did heroic experiments with 2 photon targeted patch on these, olfactory receptors I showed you in green and red, the beautiful image with the axon. So what they did is they went to this image, okay, and they patched from these dendrites in vivo, okay, from these dendrites of the axons of the dendrites that go into the olfactory receptors, and describe the receptor fields of the neurons responding course, they did not do a patch of the axons, but they did this in the Petri dish. So that neuron paper from 2010 is about doing this indirect comparison of the ORNs of this mouse that are expressed in green. And what they show is they did a screen of a smaller number of screens, but nevertheless they found that this OR responds to 7-octanal and another odorant, 006 gal, whatever, I don't know what this odor is. Okay, but the strongest response are for this particular odor. And when they recorded from the microcells that go to this glomerulus, what they claim is that this peak odorant is actually octanal. So he says, we go back again. No, it's faithful transmission. The problem is, or the secret is that this is not a real M71 mouse. Okay? It's a swap mouse. Because we did not know at the time, or it's 2010, what is the ligands for M71. Another group made a mouse where they stole an olfactory receptor from the rat called I7, which does not exist in the genome of the mouse, and they cloned it into the locus of the M71 receptor, so it would be on the dorsal side, and we could patch it. So basically, people say, it's not the endogenous receptor; it's something that you stole from the rat and you put in the mouse, so it's not the real thing. So we're still debated at the level of the mouse. Okay, 2010, 2013. This is another paper by a group in Texas. They do something else now. They inject the calcium dye into a single glomerulus. Okay. And they look, so this is shown schematically here, this is just the real data. They inject the calcium indicator, a synthetic one, into glomerulus, and they get the microcells that respond to this, that connect to a single glomerulus, and they do calcium imaging around the cells that send their axons here. They didn't do the olfactory receptor neurons. Just the neurons around the periglomerular neurons, these inhibitory neurons around that, and the microcells. And what they show is that if you look at how many odorants does a neuron respond at the given levels, okay, there are 3 different levels of neurons here, what they claim is that there is a sparsening of the response. So the opposite of what Rachel Wilson showed in this figure where Todia didn't like it, okay, where the receptor fields became larger, but something interesting happened inside. So they say actually there is a sparsening of the receptor field. It's not directly measuring the olfactory receptor neurons, but nevertheless, this is the claim of the paper. So we're still debating. And our group did something else using calcium imaging of the sort that you did in the methods group. So some of you came to my lab and did this type of imaging, right? Looked at So this is classical imaging that Yoav Adam did before he left the lab. Okay, just plain odorant. I'm not showing it, but these are all the microcells, and you had a feeling of how it looks like. I think you worked with Amit and also looked at the microcell response. But what we did in this paper is to correlate inputs and outputs. So basically, we screened several inputs, several odorants, and did the intrinsic signal imaging to find the maps that different odorants induced, and looked at the correlation of the maps, and therefore we could say some odorants look more similar to others. So for example, ethyl acetate, propanol, and propanol have high correlations because they activate similar glomeruli, so we constructed these correlation maps. And then we asked whether microcell activity of the population, as a population response, also shows this correlation. So as a population, can we see similar responses between ethyl acetate and propanol, or butanol and ethyl butyrate, which also show very high correlation? And what you can see is that the correlations drop at the microcell level. So you look at input correlations, output correlations, but then they drop, and then you can just plot the ensemble correlation of the microcells, to the similarity of the intrinsic signal image, and you can see that this is not a high correlation. What was interesting in this paper is that we looked at other cell types where there was very high correlation.
This is the correlation between what?
This is the correlation between, between these pixels to these pixels in the middle. Okay? So we have neurons with very high correlation. Okay, so let's for example look if the olfactory receptor neurons have very high correlation between butanol and ethylbutyrate or butanol and pentanol, you see these squares in the middle, then you would expect that microcell ensemble correlation as a population will also show this correlation. There is something in these orderings, but not here. So we also claim that there is some kind of sparsening. Yes,
No.
you're not very happy.
No, just we got the data when we did this analysis exactly, and we got in microcells the graph of the oral end, like the same two couples, the two propanols and the two butanols.
Ah, really?
Yeah, correlated in microcells.
Okay. Well, I would not trust your data as of the end of the course, but
Yeah.
anyway. Okay? So both the Kikuta paper in the back and our paper shows that there is some, at least in our paper we just claim that there is some kind of decorrelation of the input. Yes?
Just a background question. So in flies you have projection neurons, and in mice and rats you have microcells?
It's
Is
the
that
same, it's just a different terminology, the same thing.
There's no morphological difference or difference in the...
There is some differences, but conceptually or by design of the system, no, it's the same. There are some differences. We will talk maybe about some of them. Yeah.
In the other case, you image from the glomeruli and the microcells, the cell bodies?
The cell bodies, yes. In this case,
And
the
how
cell
do you know
bodies.
the glomeruli is only from the...
I don't know. I don't have the one-to-one match. I just do a general, we image like 5,000 or 3,000 microcells and ask how is their ensemble correlation to the different odorants. And some odorants you have higher correlation, but in many cases there is a drop in the correlation as compared to the intrinsic
How,
signal.
how can you explain the opposite direction, not the drop, but the increase in the correlation? Because you have squares here that are, if you go into the microcell, then you see a higher correlation than you saw earlier.
Here, no, we normally don't see higher correlation in the ISI similarity than here. Okay? Normally.
But how can we explain, for example, the third box from the right and the third row?
Third box from the right, on the third row, this
No.
is all one.
From the right, like two.
Here,
Yeah.
that this is lower than this
Yes.
case. I don't know, maybe it's within the noise of the system or something, I don't know. On average, again, the issue of this paper was to look at different cell types. One cell type is the microcell. Some, the correlation was very, very high, meaning that they transmit faithful information. Microcells, on the other hand, something interesting is happening here. There is a decorrelation in what's coming in. Okay. Now, when you look at the literature, etc., or the anatomy, the argument is that there must be some kind of interaction. Okay? There's so many neurons there must be some kind of information transfer. Something is happening there. And just by looking at the anatomy of the cell, of this microcell, you see it sends a single axon to the glomeruli, and they have these lateral dendrites which actually go quite wide distances. So they cover few tens or few dozens of glomeruli all around. So again, by the anatomy itself, and because you have many, many interneurons here, it means that something is happening. There's so many neurons here that they must contribute something. And just by looking at this, the first paper that I'm taking you back now to 1995, the first claim by the Japanese group was that there is classical lateral inhibition in the system. Okay? There's an input center and surround inhibition. So it exactly like the classical lateral inhibition in the retina, for example. Okay, and it was based on the anatomy, and it was based on this single experiment from 1995. The whole field is talking, or if you were taking this course when I was teaching it, 7 years ago or 8 years ago, before the H and Wilson just started to, we started believing those, that was the classical model. And you may still find it in textbooks. It's called the Mori model. And the idea is that there is narrowing down of receptive fields based on this experiment. And now you maybe have a feeling of all the flaws, inherent flaws in these types of experiments. Because, if he looks at odor space and what he chooses is the aldehyde, is the different carbon chain aldehyde, that would be a linear response as far as he's concerned. However, you already know now that that this chemotopy, if you will, is not really very strict when you look at the fine-grained structure of the olfactory bulb. But this was the first experiment that really challenged it, or put up this dogma in the air. It's a 1995 paper from PNAS, and what you see is these extracellular responses from micro cells. You can see it's coupled to the breathing cycle, which on the bottom, these neurons spike, okay? Here he puts up the odor, and when he puts up the odor, he puts up all these different odors in this case. And you can see a very strong response to hexyaldehyde and heptyaldehyde. Interestingly, you see a decrease in the response in the octaaldehyde. So there's inhibition here. And you can also see inhibition in the valeroaldehyde and the butaaldehyde. So there is strong on-center and what seems as an inhibitory surround. And when you go far enough, it becomes normal again. Okay? This is called Mexican hat decorrelation, or classical lateral inhibition. Okay, you probably know this from other courses of computational neuroscience that you may have encountered, but it's a classical Mexican hat decorrelation. So center on there's a hot spot in the middle, and there is lateral inhibition on the side. So the way he explained it here is, again, it's called the Mori model, which is classical lateral inhibition. Let's say these are the receptive fields based on carbon channel number, and now we all know again that this is not, the chemotopy is not as strict, but nevertheless, 1995, remember, this is data from rats. Let's say the olfactory receptor neuron, shown here in the gray, responds to this kind of receptor field from carbon chain 3 to 7. The white ORN responds from 5 to 9. This dotted line olfactory receptor from 6 to 11, and they have these receptive field curves. But if you look at micro cell B that sends information to this receptor, okay, to B, so this is receptor field of a micro cell that responds to this receptive field, you will see an on-center excitation, okay, based on 678, which is the strongest input that these cells receive, but you will see an inhibition from the side because these ORNs, they have a center excitation here, but they will cause this lateral inhibition through the granular system. 1995, okay, so this is the classical Mori model of lateral inhibition. This model, of course, has some predictions that were later started to be tested and broken or splashed one after the other.
I'm not sure that I fully understand what just happened here.
I'm sorry. What happened is that I explained this slide, and I have the full right to do so from the power given to me by the Hebrew University to teach this slide. Yes,
No, you
I'm
can
just kidding.
absolutely teach. I'm not
kidding,
sure that
yeah.
I understand. Just basic. So
No, this is normally new. I don't understand what happened here. Good.
the spiking diagram on the left.
Yes, that's the raw data.
Okay, so we're saying that
Recording from a single mitral cell
okay,
to all these odorants.
okay, and and we're so what the response to the octaldehyde is the
To hepta and hexy, these two odorants give the strongest response. That's the window of activation.
okay.
yes, you see that it's inhibited here next to the center, so there's center activation and surround inhibition, because around it here and around up here there's some inhibition. So there's inhibition actually from C3, 4, not so much here. This is probably still the leftovers of the excitation curve. So you see inhibition here and inhibition here, and here it resumes back to normal roughly.
Each of those bursts, is that a different kind of stimulus?
No, 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 no. You only see a single repeat. So the neuron, if you patch a neuron or record from a neuron of a mitral cell, it will be locked to the sniffing cycle. Every time the animal sniffs, inhales, you see a burst. And then he turns on the odorant after the second sniff here and records the spiking activity during the stimulus activation, and you see that a strong response here. There's just more spikes here than before. Okay? This is the spiking window is here.
So
The active window, the onset is here.
so the stimulus is an oxoaldehyde stimulus?
Here it's an undicetylaldehyde stimulus. Here it's each one is a different stimulus. It's a single neuron. Okay? And this is shown diagrammatically here. I'm patching D, and I'm now activating it with all these odorants from C3 to C11. Okay? And what you see in B here is actually what you see here in the plot, what he claimed. Strong response to 6 and 7, and a little bit to 8. Okay? No, not in this case, maybe. No, he gave another example. In this case, the 8 actually was strongly inhibited. But that's what generally what you see. Responses here are from a single neuron to all the odorants, and what he claims is that there's classical lateral inhibition. Yeah.
They also recorded from A and C, or it's just
No, no, no. Only from D.
so they just assume that
Yes,
ABC
yes.
was
They assume ABC, and they record only from D. Okay, everything is clear? Yeah.
Just a basic question, I think. Why when there is increase in firing rate, it's interesting, and in decrease is not interesting?
It's very interesting. Why
No,
not?
because the tuning curve about the substances that are increasing, right?
Mhm.
Why wouldn't I couldn't I think that this actually is neuron that senses 8 and 4, for example?
Ah, I see.
like the visual sense.
Yeah, yeah, yeah. It's absolutely possible.
It's also information. So
Yeah, 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 yeah. Absolutely. It's particularly information because there was activity before. Yeah. Yeah. Absolutely. You can say the tuning is upside down. But normally, yeah. But I'll let you know why later on it's a little bit more complicated. Okay? But generally speaking, yeah, it's a little bit awkward design, I think, but you know, fair enough.
it's awkward for us as humans, but it's
Yeah, 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 but I agree. I mean, inhibition is as important as excitation, especially if there is, you know, on activity. If a cell is silent and then you silence it from zero, it maybe is not as efficient in terms of the, you know, using the dynamic range to its maximum, but I hear you completely. I'm with you completely. Again, I'm giving you the ABC of attraction, okay? Just so we know where we are. So the predictions there are several predictions. First of all, that receptor fields of mitral cells should be narrower than the orange. That's the first prediction. And as you already saw, this is highly debated. So good people like you, spend their best years trying to look at this, you know, so you end up with a nice neuron paper or a Nature Neuroscience paper, but this is still a debatable issue, certainly between mice and slides. The other prediction, if the MOE model is true, is that you will have a classical center surround inhibition. So a center on, and the inhibition will fade away as you go away from the center, from the on center. Okay? That's one thing. The other thing is that if it's like this, because this is actually the brain brain comes in 2 dimensions, in 3 dimensions, or in 2 dimensions, if we plot the glomeruli up here, you will actually expect to see more inhibition than excitation. If you patch randomly, so inhibition will be much more dominant than excitation if the Mori model is true. This was actually, again, challenged and found that it's not the case. Actually, in this paper by Ian Davison, what he did, he did the largest screen to date in 2007 of odorants. He used, I think, 250 different odorants to map mitral cells. It was actually in the lab where I did the postdoc. Ian is a good friend, so he would take a mouse, patch a neuron, and he built a robot of 250 odorants, and he read the newspaper. So he would come to the lab every day. He comes early at 8. By 9, the electrode is in the brain, and he reads the paper, and this robot goes, screen 250 odorants to see the receptor fields of the mitral. Okay, what Ian found is that actually he has the same amount of excitation and inhibition on average. So the same number of cells are excited and inhibited. So that was a very strange result if you consider the Mori model as well. So there's no significant overlay inhibition. It's not dominated by inhibition. Okay? So suggesting that there is no massive lateral inhibition, or at least not in the design that Mori taught us in 1995. And more explicitly, later on, a year later, the group of Marcus Meister, the same one who crashed the 1 trillion other papers in this mathematical archive paper, explicitly tested whether this center surround inhibition is true. He said, okay, not only do we expect more inhibition, but we expect it to be stronger close to the on center and fade away as it goes away. What actually he found, he used photoactivation, optogenetic photoactivation. They use fancy mirrors to activate the different glomeruli by patching a single neuron and now flashing the light on, like in a pattern. So they patch a cell, let's say here, and they start flashing lights all over the brain with optogenetic activation. And what they found is actually this scenario. So it's true, every time you patch a mitral cell, you get an on-center, because its dendrite is up here. So you get only a single on-center, and you get on average only 4 inhibitory centers. So you do have a little bit more inhibition than excitation, but it's never in a nicely structured form. So it's not what we know from the visual system, but rather there is an activation pattern which doesn't fall to the classical Mori model as it was taught in the textbook. Yeah. So when he flashed his light here, he saw inhibition in the mitral cell. And when he flashed his light here, he saw also inhibition. Here actually the strongest inhibition, for example. Okay, we won't go into the data of this paper, but just what we want to, every year I give in the exam the question, what is the Mori model and what are the evidence against it?
What is it?
Okay? Not this year, apparently. Yeah.
So in the Mori model, the lateral inhibition is caused by the fact that it is connected to the nearby glomeruli by inhibitory connections, right?
Yes, these granules will connect.
So if I touch the mitral cell directly, why would there be a lateral inhibition if it's not through the glomeruli?
What do you mean?
Like in the model, the glomeruli would stimulate, would excite one.
Yeah, he
one
will
mitral
excite
cell.
this glomerulus, because in this mouse all the olfactory receptor
Oh,
neurons.
this is in the glomerulus level?
No. But it starts from the glomerulus, right? Because normally, under normal conditions, when you excite an odor, things are much more complicated because you get a pattern of activation. So when you give an odor, you'll have 7 glomeruli that like that. But Meister did the very basic connectivity scheme. I'm patching a single cell, and I'm going glomerulus to see what its effect on this mitral cell, just to get a connectivity structure.
What about if it's like the actual natural thing would be like several?
Natural thing is much more complicated.
So when it's like just one neuron to inhibit it slowly. Yeah, no, this I get, but
Yeah.
I mean, it's just, it's very, like I would imagine it's not, over here it's like you said, you activated one point at a time and check
Mhm.
what happened.
Yeah.
And I would expect it to be like five, like more than one neuron that are activated at a single point has their effect on, like,
Yes. So
maybe
what you
the,
say is that, well, first of all, what he claims, this is this landscape of inhibition is this and not otherwise. What you say in terms of the odorants, there are odorants, many odorants that will
mhm.
excite this glomerulus and therefore will inhibit this cell. But on average, when we do, if that's what you're saying.
Not quite, but okay.
Okay. But that's what I mean. So one expectation would be that there's actually going to be many more inhibition, much more inhibition onto the microcell, and in fact,
So
based
this
on
is
the previous
actually the
paper,
minimum.
what?
This is the minimum.
The minimum,
Right?
yes. But we do see that it's largely balanced, the amount of inhibition excitation a neuron receives.
I don't understand why would we expect an organization like in B, because in the eye, for example, there's the importance to the actual mapping of the retina, right? The location.
Yes.
In the nose, I would expect it to be very salt and pepper. So if you have an injury or something.
I like what you said, that's fine. We just published a paper why maps are not important in the chemo world. Yeah. No, there's no reason to think about it, but that was the dogma in the field. Again, people who graduated, 7 years ago, 8 years ago, thought, the exam is lateral inhibition in the olfactory system. And as we go along, we kill this dogma one by one. Yes.
Here, I just want to see if I understand the experiment. They shine light on the entire.
They catch this cell,
They shine
and
light.
they shine a patterned light each time on a pixel in a
What's the
very.
pixel? Is it entire glomeruli? It's a lot.
It's actually smaller than one glomerulus. They use also genetics on I don't know, 100 micron square, and they do it systematically. So actually they do it in a random fashion. It's a very nice experiment. It's like a random dot, and then they do backward spiky line, and they get strong inhibition on the flash was here, strong excitation, and this type of inhibition.
So how many inputs they trigger in every time they do?
Every time they claim they manage to make a single glomerulus. That's what they say.
Okay, single glomerulus, but how many inputs are there in this single
Is it
glomerulus?
successful?
About 15,000 axons coming in, exciting a single glomerulus.
You're exciting all of them at the same time.
You induce a strong activation. When you change the odorant concentration, etc., also you induce different levels of activity.
But when you give order, I wouldn't expect that it will be 0 and 1. I think
It will be 0 and 1 for one glomerulus,
yeah.
maybe, but much less to others.
Yeah, but in the case for a single smell.
No, it's not 0 to 1, it's between 0 to 15,000 neurons that are activated.
Okay, so maybe there are in this 15,000 subsets which are inhibitory and subsets which are excitatory.
No, in these 15,000 cells, no, it's all excitatory projections of the ORA.
No, but from the glomeruli,
Yes.
glomerulus to the microcell.
Ah, fantastic. That's what we're talking about. Is there any transformation?
Yeah, but there is no from one glomerulus. There is no just one type of connectivity. Not one glomerulus is not just inhibitory or just excitatory. There is a balance. And
Okay.
if you will activate
If you activate, so what? No, what they have. So this is the mouse, where all the olfactory receptor neurons express channel oxytocin, but only the olfactory receptor neurons. So all the ORNs that come into here, at all glomeruli, have channel oxytocin. And then they flash the light and they activate, excite the olfactory receptor neuron of a single glomerulus. That's what they do. This in turn causes excitation of inhibitory cells, etc., and causes maybe lateral interaction.
but it's not physiological. So how can you say that
No, it's not physiological.
other glomeruli, even though you do not see that they're inhibiting,
Mm-hmm.
it doesn't.
No, you do see they're inhibiting.
No, other that you don't see.
When you excite this, you see no effect on this microcell next door.
Because it's like all the neurons.
You suspect that you excite all the microcells that are, or most of the microcells that are connected to this glomerulus. And based on Mori, we actually expect the strongest inhibition if you will excite its next door glomerulus.
And if you go neuron by neuron and not just glomeruli by glomeruli, maybe you will see that the organization is different.
Yeah, okay. So that's a good point. So next, not in this slide. In this slide, they looked at neuron by neuron, but in slices. So they make a slide, and then we go out to, we'll take a break. So they patch two neurons in a slice next to each other. And if the neurons are projecting the dendrites to different glomeruli, they never see an excitatory response. Sometimes they do see inhibitory response, but clearly not a nice projection onto the cell. Okay, so this is an example of two neurons which they patched in a slice with all its issues that you slice many of the connections. They never see strong connections between, or in many cases not strong connections, and they're never excitatory. But in the slide with Rachel Wilson, when she did, you remember she recorded from this and this to show how receptive fields widen. But now, in another paper from 2007, it's a nice Nature paper. It's not the paper you're doing.
It might be.
Maybe.
No, I think it is, yeah.
So she did many combinations, but when she patched, she made the slide with only one receptor intact and patched its next door neighbor. And what she sees is lateral excitation, which explains the widening of the receptor field. Okay? But in fact, they found this companion paper in Cell. Gero Meisenbach found the source of these cells. It's actually huge interneurons that have synaptic connectivity all over the antennal of the slide. So the dogma to date now is that we still did not find lateral excitation, although I think one of my students found some lateral excitation. We're not sure yet, so stay tuned. But in slides there is lateral excitation very clearly, and this Mori model is classical in the field, but not completely explained. So this is a modeling study, and which shows this Mexican has the correlation, but he has different explanations to it, and we'll talk a little bit about it after you come back from break. Wow, funny.
יש לתאים,
בדקתי אותו, אז פה היה...
מה שחור היה ND. וכל השאר לא היה כאילו ND, לא היה כלום.
לא, אבל איך אתה יודע, איך אתה יודע את מה אתה הערת ואת מה אתה מדדת?
לא, אני תמיד מודד את האמצעי, וכל
אתה
פעם
תמיד
אתה...
מודד את האמצעי. נכון. אז שהערתי עליו, זה היה מעט ND. איפה היה 40? איפה היה 40? כל השאר לא היה ND.
סתם, השחור זה אומר שכשהערת פה, אז קיבלת הרבה ND. השמן זה קצת מבוצע,
זה אותו זהב, כן, בסדר. ואז כאילו, ולפי המודל של מורי זה היה אמור להתפלג כאילו...
כן, זה היה צריך להיות בסוף, זה שקרוב יותר, אבל אתה עושה יותר קצת, כי זה יותר קרוב לטלוויזיה, ואז כאילו...
Infinity stuff?
Infinity?
Infinity?
כן, זה היה צריך להיות כאן לבן, זה היה גרפים פחות לבן, כמו גרף הכללי.
כן.
אהבתי את זה, שבאמת יכול כאילו להגיד איזושהי תיאוריה,
ושנים
צריך להפנים אותה, כאילו, אבל אני לא כאילו סתם כל... אתה יודע, מדד, אמרתי לו, מדד איזשהו מייקרוס איזה אחד, ולפתח את התיאוריה.
רגע, מה, הוא איזה ניסוי עשה?
הוא רק מדד מייקרוס אחד, והוא הראה שהוא יותר, שהוא רגיש יותר לאיזשהו...
מייקרוס זה התחנה השנייה כאילו.
כן.
אוקיי, אז מה הוא? זה מדד ומה?
והוא ראה שפשוט יש לו, הוא יותר רגיש לאיזשהו ריח ספציפי. זה פה, זה התיאוריה.
שמה?
שהוא רגיש לריח. שיש לו אקסיטציה רק בריח, כאילו יש ריח שבו יש הרבה אקסיטציה, ובשאר הזה יש נדיבות.
במייקרוס זה שניים יש נדיבות?
לא, באותו סל, תן לו אורגר כזה. זה מה שהוא עושה. ואז הוא גילה שכאילו הוא רגיש, מאוד מאוד רגיש רק לאיזשהו ריח אחד, ולריחות אחרים הוא לא רגיש.
הוא עשה את זה כאילו בתוך אין ויבו או זה...
לא, לא יודע. מייקרוסרה, אבל לא יודע.
אז איפה הוא עשה את זה? איפה אתה יודע שזה סתם כאילו...
לא יודע.
יש לנו, אני מנסה לשתף פעולה עכשיו עם דייסורות, עם קר דייסורות. גם ביחד.
מה מוריס אמר שהמצב אצלם?
קודם כל הוא חלק מהמקס-פלנק תזות האלה, הוא התחיל רשמית. והוא אומר שאחלה, שבנו להם ומתקדם, ומאוד מבסוט. סודי. זה שאני מקריא.
זה יכול להיות שמקליטים?
בדיוק. תסתכל. זה אימג'ינג של כל המוח. אשלח לו דוגמאות עם הסינגל ניורון שיעשה את כל האקסונל פרוג'קטים.
זה כאילו התחלה?
זה שיטה חדשה, על בסיס פסיכולוגי, איך קוראים לזה? קולמן, קולמן.
מה אתה?
אותי מעניין projection patterns של סלים בירטים. אני מנסה לראות האקסונים שלהם נופים. כרגע אנחנו מחכים לאט לאט. אני רוצה לעשות את זה שיהיה לי קצת מחקרים. אז I'm into anatomy. אבל מוריס אמרו יותר פעולה.
אלכס אינשטוק? פשוט ראיתי שולחים באלסייק ניוזלטר, אלכס אינשטוק, משהו בנייצ'ר, שיתוף פעולה עם ג'ף ליכטמן.
כן? זה עבד להם עם הבוין באוזן? לא יודע.
משהו כזה, כנראה, משהו שלראות גם.
אני חושב שהם עובדים על
סינגל אקסון פאטרן.
אני חושב שהם עובדים על ה-peripheral nervous system.
זה לא ממש קראתי.
לא ראיתי. אני יודע שהוא משתף את הפעולה.
אוקיי, הנה, אלכס אינשטוק עובד על כאלו.
כן. לא, אבל הם מכניסים כמה צבעים לתוך גנגליון כזה, ומסתכלים האם תאים שולחים ל-divergent או convergent.
אוקיי. אז גם בנושא הזה, נדל, של מונה.
כן.
אני אפילו לא יודע אם מי זה, אבל מישהו בניו יורק ששולח להם עכשיו תמונות שלהם גם כזה, all brain או all cortex.
כן, יש כמה קבוצות. לא, ראיתי אותך, ואתמול פגשתי את מוריס. אתה משתף אבל פעולה עם החבר'ה בגרמניה.
כן, בינתיים עם מרסל.
כן.
ויש לנו דיברנו עם מוריס שמתי שנדענו על זה.
כן. הוא רוצה לשלב בין ה-long range connectivity וה-local connectivity. הוא מסמן אותם מרחוק, ואז הוא עושה reconstruction בעיר של קוביות.
זה מאוד רוצה להוסיף איזה כמו של long term.
long range.
כן, long range.
להוסיף long range לתמונה, ואז יש פה סימון זה משהו שאותי מאוד מעניין, אבל ברמת ה-light microscope, אנחנו עושים כמובן. אני עושה, very artificial, אני עושה long range staining, ורואה איזה מבחינה פונקציונלית, איזה אינפורמציה הולכת לעבוד.
אתה רוצה layer to peer מפה? אתה מסתכל כאילו מי שולח
dream experiment, יש לך פה נניח קורטקס, נניח זה layer 2, יהיו תאים ב-layer 2 שחלק ישלחו לתחנה אחת, אוקיי? maybe some of them לתחנה נוספת, חלק רק לתחנה הזאת, חלק לתחנה שלישית, אוקיי? אז אנחנו עושים forward anatomy, שיטות כאלה של לראות מי מדברים. Once we will find that, אז אנחנו מסמנים אותם retrogradely, כי אז רק התאים האלה ששולחים לפה יהיו צבועים באדום. ואז אני עושה two photon imaging, בחיות מתנהגות, ורואה מה ההבדל בין האינפורמציה שהולכת לפה, לעומת האינפורמציה שהולכת לפה. כי כרגע, everyone is equal. אתה מדבר עם מרסל ומדבר עם אנשים שעושים אותם מהמחלקה, nobody is equal. There's high heterogeneity. We don't understand how the information flows in terms of... אז אותי מה שמעניין זה in terms of final connectivity or destination. לא, אבל זה מאוד קשה לקבל...
מרסל גם עושה את זה כמה דברים כאלה.
זה מאוד קשה לקבל specificity, ועכשיו יש לנו אחריות שאנחנו... יש לנו high specificity, וזה משהו. שהם רק להרטיב, מסתכלים מדי פעם ספורה. עכשיו we're tracing from them, we're silencing them, doing Davis tracing, input pattern, וכולי וכולי. רק בלהרטיב. לראות לכמה מהתאים שלו מסומנים בלבן. כן.
מי מסומן? אני לא...
מסומן התאים ב-Layer 2 לעומת התאים ב-Layer 3 שהוא מתחתיו.
אבל לא בתוך...
לא, ב-Layer 2 אנחנו, לא יודע אם יש לו מספיק תאים, בדרך כלל זה 30 תאים בכל קבוצה. לא נראה לי שיש הבדל משמעותי. אולי נצליח לדבר בשנה החמישית של המקס פלאנק, אנחנו נצליח ליהנות מהעבודה הקשה שלכם.
אני עכשיו מקבל בסוף תוכנית הקשרים ריבועית. עד עכשיו אני לא...
בוא נקבע עכשיו, לא תוכל להסביר.
לא, שלחתי מייל.
לא, שלחתי מייל, פשוט חזרתי לשתיים,
כן, זהו, רק
בלילה, ונחתי באמצע הלילה. טרם מספיקותי לענות לכל האימייל האדמיניסטרטיבי. מה? אני לא עשיתי כלום, אני סתמי.
זה לא הכל נכון, זה אוויר, לא היה כאן חלום. Apparently, I'm the minority. We don't know. No, Dean, you've been a great partner in this battle for air conditioning, temperature.
Okay. So again, we go back to this lateral inhibition, and the idea is that the inhibition is the more we go into the system, the more we see that there are probably interesting things going on. Tom Cleland from upstate New York, Cornell. From Cornell University is a very a good computational scientist or theoretician with some nice ideas. So he is modeling all these networks. So in the context of this lateral inhibition, and the idea was that the underlying receptor fields of mitral cells overlap considerably, and this inhibition is helping for contrast enhancement between the different glomeruli. So that's one idea, and you can get it by different means. You can get it by this Mexican-Had decorrelation, which we talked about until now, but you can also get it via non-specific inhibition. So if the mountains they have a significant overlap, if you decrease, just do a non-specific inhibition, you can still get an increased contrast. The more modern version of these things is actually all kinds of functions that are affecting the output neurons. Okay, so if you look at the output neuron, it's not a faithful transmission, input-output, but rather these different interneurons in the olfactory bulb contribute some form of gain control onto these cells. Okay, so they can amplify, they can inhibit, and this inhibition can take different forms. And we will talk about a few of these forms. So if you just look at the input-output function, which is all these graphs are basically input-output functions, like we saw from Rachel Wilson's olfactory receptor neurons to the mitral cells or projection neurons, which you saw last time. And they take this type of form, okay, these input-output functions. This is when you plot it linearly. If you plot the same graph, the same type of input-output function in a log scale, you get a response which looks like this. And now, if we now start to sink our teeth into the circuit and try to understand what each type of inhibitory neuron is doing in the circuit, we find different examples of different gain functions. For example, let's say a given inhibitory neuron causes this type of change onto these cells, okay? And we can do this by photo-inactivating a given cell type or activating a given cell type. In many cases, when you will record from the microcell the input-output function, you will get this type of effect, and this type of effect is called input gain control, and you are going to talk about it.
Yes.
What?
Yes.
Yes. Okay. So we're going to see an example of input gain control, actually from the presynaptic olfactory receptor neurons in a paper by Wilson. Other people, like myself, and I may skip it, we found output gain control on the microcell, okay, from a different inhibitory cell type, because when you plot the input-output function of a given cell, you see this type of an effect, okay? Actually, when we inhibit the cells, we saw this type of effect, which is a linear output gain control on the cell. Other people in zebrafish, then last year I asked to give a paper in class about another form of transformation which changes if the inputs are weak or inputs are strong. This is actually called dynamic range compression, which is something that you actually find in video cameras. It's a filter that you find in video cameras, which has an opposite effect on the weak inputs and on the strong inputs. Okay, so it causes this type of effect onto the circuit. So basically, if you now join a lab of olfaction or read the modern literature, this is a paper from 2014, we now understand that each subpopulation of cells in the olfactory bulb contributes a different form of gain control onto these output cells. Yes?
What is exactly the output? The firing rate?
In this case, it's the firing rate. And it gets more complex. We will hopefully go along. This is things you will touch upon, the synchrony, for example. Okay? But if you look at a single input-output on a single cell or a single glomerulus, you already start to see the influence of the computational neuroscience community on the field that, what exactly is happening there. It's not a faithful transmission. And the reason is that there is a huge amount of inhibition in the olfactory. So normally, if you look at the cortex, you will see 80% excitatory neurons, 20% inhibitory neurons, and people study a lot the excitation-inhibition balance and things like that. In the olfactory bulb, we have 99% of the cells inhibitory, and only 1% of the cells are mitral cells. So although you look at the textbooks and you look at the classical papers by Axel and colleagues, they normally look at the olfactory receptor neurons and the projection neurons. But there's a whole lot going on in terms of computational power of this structure that is only starting to being explored, okay, if you will. Rachel Wilson was perhaps one of the leaders in this field. Having these genetic tools, you can now these are not all the same. Each one, I just told you there are granule cells and periglomerula, but actually there are many cell types. Okay? Let's jump here. This is just an example of real data. So if you look at, this is something I give in my talks a lot. Just look at an immunohistochemical staining of only 7 inhibitory different cell types. The glomeruli are up here. This is one glomerulus, another glomerulus. These are the cells that surround it. So different cell types, the periglomerular cells have different cell types. These granule cells have different cell types within them, some with these molecules, some with these molecules, etc., etc. We, in my lab, Yael Shlomai, study these PV cells in the middle because they are very distinct molecularly and also in terms of the spatial region, okay, so they're not intermingled with other cell types, but rather very distinct, and we studied the structure and function of these cells and found that they are doing an output gain control on the mitral cells, which sit down here. But in order to really understand the mechanism of computation in the bulb, different cells do different things. And again, I want to run maybe fast. Yes.
All interneurons are inhibitors?
Good question. No. But 90% of the cells are inhibitory. There is one class of excitatory interneurons. We know nothing about it, but we have the mouse, and in 2 years I will know what they do.
We just talk about mice here, right? Because the Drosophila, I know that the T-cells can go.
Yes, Drosophila also have lateral excitation and inhibition, but we will. It's a little bit different. It's different because there it's small, and the interneurons in Drosophila, they actually send their dendrites and axons all over. So they have these global effects.
So now we just talk about mice.
Now mice. Yeah. So we don't have time to tell you about Rabies tracing of these cells and the responses. These are our papers. But different people have. I'll skip my papers, although it's easier for me to talk about, but never mind. Well, maybe just a word. Just a word. So basically what we did is we took these mitral cells and asked what is the synaptic connectivity onto these mitral cells. Okay, so you take a mouse which has Cre only in the mitral cells, and you use a method called Rabies tracing, which we actually imported to Jerusalem and is alive and kicking here as of last month. And then you can ask what are the presynaptic neurons to these mitral cells. In this case, it's these yellow neurons, and then you can plot where do they get their input from the olfactory bulb. And what we find is that they get widespread input mainly from the PV cell. And then we looked at the synapse. The synapse is very strong, and our colleagues did this experiment of before-after. When you take out the PV neurons from the circuit, this is back-to-back paper, the neuron, and what they see is this linear gain control. Anyway, so that's the PV cell. Cleland, the theoretician, in 2010 actually said that there is an inherent inhibition in the structure of the PV by the periglomerular cells. Periglomerular cells are very small, their input resistance is very high, and therefore they spike very strongly for very weak input. So if you look at the glomerulus, maybe open that. He says there is no lateral inhibition at all needed in order to get contrast. Okay? So there is this glomerulus, okay? They get input from the ORNs, this you know everything about, and mitral cells are here. But inside, so there's a strong synapse between the ORN, an excitatory synapse between the ORN and the mitral cell, but there's also these periglomerular neurons which receive input from the ORNs, okay, and make an inhibitory synapse onto this mitral cell, the red one. So this is basically what's shown in this diagram. Okay, so this is the olfactory sensory neuron terminal, and it makes a synapse onto the periglomerular neuron and the mitral cell dendritus. But because these cells are small, their input resistance is high, and they fire very easily. So when a weak odor comes in, these periglomerular neurons start firing already. So when an odor comes in, the axons of the ORNs actually excite the inhibitory neuron first, and actually you will see an inhibition onto the mitral cell. And there's no lateral inhibition at all. It's built in by design of this glomerular inhibitory structure. So if you give a moderate input, he actually finds that the PGNs will fire strongly and the mitral cell will be inhibited. But only when the input is stronger and stronger from the olfactory sensory terminal, then the mitral cell also, this excitation will override the inhibition the mitral cell gets, and the mitral cell will fire. Okay?
You say it's only theoretical? Would anyone test it?
Thank you for the question. This was not an invited question. And last year, a Nature Neuroscience paper from 2010 2014 actually tested the different contributions of periglomerular neurons and granule cells.
You can pick them out, like genetically, specifically
Uh, Sort of.
Okay.
Okay, so this is the paper. I actually read it. I put it in because I read it last night in the airplane, and I thought it fits well to the story. Basically, what they do is they use optogenetics to silence all the granule cells or silence only the periglomerular neurons. And they patch microcells and they silence the inhibition, if you will. Okay. And what they find is that the granule cells don't. The granule cells, which Maury talked about, you know the connections on the lateral dendrites, actually don't affect at all the inhibition onto the microcell. The only effect that they find is when they silence the periglomerular neurons. So then when they silence them, they see that there's a rebound of an effect.
Like this, so maybe they silence the glomeruli as well because it's not isolated?
No, they have good controls. I mean, it's not too bad. The problem is that they silence many cell types around the granules. But this more striking thing actually is their negative result, is that the granule cells don't affect the inhibition at all of the microcells. What the granule cells do, and this connects to your paper, is that they affect the synchrony of the responses of the different microcells. So when they silence the granule cells, the shift in spikes, the accuracy of the spikes within the breathing cycle is changing. Okay, so normally these spikes, as you saw, they fire in bursts, which are locked to the sniffing cycle. And when you take out the granule cells, the cells become less coherent in their spiking synchrony. Okay, so this is a paper that came out in December last year, so it's fresh. They talk about all kinds of gamma oscillation, which affects spike timing, which is important for, people say it's important for odor coding. Okay? And I think the third paper, the Schusterman paper, not Schusterman, the smear paper. Who is doing the smear paper? No? Really? No, we have the smear paper. No, Tamar is at the end and she's... Anyway, we have 3 papers as far as I know. 4 papers actually. One on the... Yeah, you're on the pheromones. Yeah. Next talk is going to be pheromones. Yohan will show. Okay. Anyway, Dima Rinberg's paper is doing very nice behavioral paper where he's playing with the timing of the sniffs and the spikes, and he shows that the animal uses this information between inhalation to the time of the sniff to decide, make decisions on behavior. That's a big issue here. Let's go fast about this. The only thing I want to say here is that one of the changes that when an odor comes in, the recent bon ton in the field, is that odor information comes in not only in increases in spike rate, so when an odor comes in, there is going to be more spikes, but the changes in the phase of the activity between the different microcells. And specific odorants will cause specific microcells to spike at the same time. And it's this synchrony that is read out by the next station that is important for the code of an odor. And if you give the same odor at different intensities, it will shift the time of the spike relative to the breathing cycle, but the phase between the different cells will remain. Okay? This is actually was brought up by Hopfield, the famous Hopfield, in a 1995 paper. This is just a review about this paper. And it's something which is still people work on, and there's debate about what is the neural code. But basically the idea is that if you want to look at a code, any code in the brain, you want to ask the next station, which has to read this code. Okay? And the next station, the claim is that they are reading the synchrony between the different microcells in the code. So that is what is changing between different odorants, is how synchronized they are. And I think you're going to talk about spike synchrony, which has information about reward or something like that. Yeah, and when I will read the paper, we will discuss it. Okay, let's skip this. Just to keep in mind, in the back of your mind, is that a big, big, big computational aspect was brought up by Gilles Laurent and his colleagues in early 2000, and that it's all a population code, early days of computational neuroscience, and actually the information of different odors come in the decorrelation of patterns of the population as a population code. So if they studied this in locusts and fish, later rhino and fish, still does, and still argues that this is the most important information. And if you look early on about the correlation between the population for different odorants, you see a very high correlation early on in the response, at least in the fish. So if you give all the different microcells which are shown in this matrix, and you say, what is the response vector for this odorant and a different odorant, two different odorants, if you look early in the response, if you are a decoder looking at the population, you see a very similar image. These are the same microcells. But as you go along the response, 300 milliseconds later, 500 milliseconds later, etc., the population starts to decorrelate and the information is in there. So there's many Nature science papers on this decorrelation, how information is gained as you go in time in the response of the population is decorrelated. Zach Mainen in 2006 poured a lot of cold water on this theory by showing that mice can actually make full discrimination at 100 milliseconds. So this whole literature was about how patterns are decorrelating after 1 second. But once Zach and Uchida showed that the mice can discriminate after 200 or 150 milliseconds, they say, it's all good and true. Computational neuroscience is great. You get all your function, you do bits of information and what have you. I don't know. You guys can calculate this much better than I do. You get lots of bits of information here. But the mouse is already, shopping in the shopping center here. It doesn't care about this at all. So the question is whether the mouse uses all these decorrelations is still debatable. Then Reiner showed that actually there's decorrelation in shorter time frames, so he's still battling this theory. And these are two opposing machanot, camps. Okay, people who think that decorrelation is more important, very computational people. The other ones who are thinking that spike, the Hopfield model of the spike synchrony, etc. Okay, we are done with the olfactory bulb. Now we go beyond the olfactory bulb. We don't know much. And what we finished today, what I'll get to is, I don't know, if I don't get to the slides, we'll just delete them from the stuff for the exam. Next Sunday, Yoram Ben Show will give you 1 full hour, 60 minutes. So that we'll take a 15-minute break and hear one seminar about a very cool paper on tears and sex. Okay? And with this we finish the olfactory system and we go, Wednesday I think we start the auditory system. Unfortunately we don't have enough time, so we'll do it very basic. Ellie will give you some more advanced, but what? Wednesday you talk, sorry, the following Sunday.
I have a question. The last Wednesday, I think we have 3 seminars, but it is on the same time that the Brainy Day concert is on. Can we just move them to Sunday?
We can talk about playing around with the schedule, but we have to give you want to give up 1 talk on the auditory. That will be hard.
Only the third,
Let's talk. Sure, I'm flexible. As long as we do the seminars and do the lectures, we can come on Friday if you want, no problem. Yeah. No. Whatever change we make will not be obligatory. The brainy days actually is very good. They're very good people coming into the from outside. Okay. So this is yet another diagram of the olfactory bulb. Now, I mean, you will be masters of drawing these this circuit diagram. Again, this is the nose, the olfactory epithelium. These are the olfactory receptors sending their axons to glomeruli. These are the micro cells sending a single micro cell to a single glomerulus with the lateral dendrite. And now we're talking about the next station. Okay? We know very little about the next station. We know there are generally 5 or 6 downstream regions from the olfactory bulb. One of the most interesting things in the olfactory bulb is that it actually projects directly into regions of the business end of the emotions, hypothalamus, amygdala, etc. And this is the reason we think it's the only sense not going through the thalamic relay. All other senses, you know, go from the retina, GN, thalamus, etc. Also auditory. Here it goes directly to the business end, to the kishke of emotion. And that's why people say, again, the hand waving, that some smells cause plasticity very early on, and it sticks there forever, and you have the smell of home, and the cake of your grandmother, etc., etc. Okay? So smells go deep, deep and basic. Okay? And there is a scientific connectivity reason for this grandmother story. Anyway, in practice, they send their axons to different regions. One is the anterior olfactory nucleus. It's a very close by station, the first one. And this is thought to integrate the different sides of the brain. We know very little about this. We talked about this just, there was a paper in 2006 called Rats Smell in Stereo, mainly driven by what we know in the auditory system of how we calculate where a sound comes from, how we can localize sound in space. And there's very, very little known. People criticize this paper quite a bit, but the bottom line is that if you give a mouse to smell, if smell comes from a given region, you'll see very strong activation in the, let's say if it comes from the right side, you'll see very strong activation first on the right side, and very little on the left. So there is lateralization. Whether mice or rats really use this information to detect where an odor comes from, I suspect yes, because they say that when they close one nostril, the rat has problems to detect where exactly it is or makes mistakes. So I guess we are using our two nostrils to compare, but we know very little about it. So, you know, basically there are 5 papers on the field, and this is an older one which made some noise, and this one is 2013. One interesting gossip issue here, many of our students, you know, they are modern and smart, etc., so they go to hike in wild places, sometimes for a year or 2, and they always want a scientist in these wild places. Upi Bala is in India. For those of you who love India, he's a top-notch computational scientist that works in India. There are very few of these in India that are publishing in very high-profile papers in no sense. So if you want to collaborate with India and Shanti, Upi is a great place to go. Yeah.
Most experiments are done like both sides. You don't block with a certain nostril or
Yes.
the regular.
Yes. Yes. Normally, yes. Sometimes there are experiments where we want to compare left and right, so you close the nostril here, look at activity dependent, but mostly from the front. This was last year or 2 years ago. Again, our friend Rachel Wilson, with a mind-blowing paper in Nature, she showed that Drosophila flies can also go detect left or right. Not as good as we are with our ears, but nevertheless the flies can do it. And she can teach a fly to, if the odors come from the left, they will fly to the left, to the right they will fly to the right. And she says it's very important because, you know, these guys fly garbage, etc., and they want to know exactly where to go, whatever. And what she shows is an amazing mechanism of how they do it. And one thing I did not tell you is that in slides, not in mice, in slides, and there's always, when you learn to examine the material, there's always this tension between what we know in slides and mice. So keep that in mind. Actually, in slides, the ORN sends an axon collateral to the other olfactory bulb. So from the same olfactory receptor neuron, one axon goes straight up, and there is a collateral from the same neuron that goes to the other olfactory bulb. And what she showed is that the same neuron has differential synaptic release of its vesicles in the same axon, the same cell. It releases differentially synaptic contact to the left and right side of the brain, which is quite amazing. It's a very basic neuroscience question whether a neuron which has an axon, if the release here in this site is the same as the release in this site. Okay? And what she shows is that the axon collaterals that go to the contralateral side have weaker release. Okay? And this way she explains the behavior. She goes on and on. Again, we don't need to go into this into the details. I just want to give you where the olfactory bulb sends its information. Okay, so first one is the AON, which is thought to be important for lateralization or spatial detection of odor. The other one is the piriform cortex. Piriform cortex is a little bit different than other cortical regions that you know. It's not a neocortex. It's an old cortex. You can find it in turtles and other evolutionary lower older animals. It's actually a 3-layered cortex, not a 6-layered cortex like we know, and it gets direct input from the piriform cortex. Several years ago, there were a few from the olfactory bulb. This is just to make sure you're awake. No. So, piriform cortex input. There were 3 back-to-back papers in Nature about connectivity from piriform cortex from olfactory bulb to piriform cortex. It's not this one actually. Let's skip this. One is an amazing paper from again, doing very simple anatomical tracing experiment, taking one of these animals, which has all the axons from the nose going to a single glomerulus, and injecting a dye into the single glomerulus, and following the axons of the microcell. Well, close the light so you can appreciate it. It's actually a mind-blowing image. So these are axons in the piriform cortex, accessory olfactory nucleus, lateral olfactory tract, amygdala, entorhinal cortex, showing axons in red down here from a single glomerulus. Okay? So only these 25 microcells that send their dendrites into one glomerulus, this is their axon. And in fact, it's unbelievable that we are in 2015. We know very, very little about axonal ramifications of neurons, where they go exactly. The few people who did this, it's Bill Sackman and other colleagues, stained neurons, filled them up with a dye, and they saw amazingly complex projections of single axons. Axons are much, much harder because it's a neuron talking to many targets. Most of what we know is, dendrites, which is easy because it's around the cell body, so we can calculate the computations here. But if you look, this is not true for other, for Drosophila and others where C. elegans, where the connectivity is fully known. We are far, far behind in mammals in what we know. Anyway, this is a 2011 paper, part of a triple back-to-back papers in Nature, showing that a single glomerulus sends its axons in the cortex all over the place. So from a highly convergent connectivity from the ORNs to the olfactory bulb, there is now highly divergent connectivity outwards. Okay? And in fact, if you record from neurons in cortex, they're very sparse. And actually, Chaim Sampolinsky and others have papers on this in Larry Abbott about the computational power of this transition in the olfactory field. What is nice here is that you can see that there is condensed axonal projection in the amygdala. This exact same conclusion was brought up by one of the companion papers in Nature, which did the opposite experiment. I'm not going to go into the details here, but what they did is they started rabies tracing from the piriform cortex. So rabies gives you the presynaptic landscape of the cell. Which neurons are connected to it. Still has problems because it doesn't give you the full connectivity. But nevertheless, they started from neurons in the piriform cortex and traced back where is it getting connections from. So basically, it's the complementary experiment to Richard Axel's experiment, which did forward tracing from a single glomerulus. They did backward tracing from a single neuron or a few in the piriform cortex.
They come from the one thing?
One synapse back. And what they find is that a single neuron in the olfactory cortex gets inputs from all these different cells around the mitral cell, the olfactory bulb. So basically, the conclusion is that highly restricted areas in the piriform cortex receive direct mitral cell input representing glomeruli that are distributed throughout the olfactory bulb with no apparent spatial organization. Okay? So input is highly accurate from the nose going into the glomeruli, and I've showed you throughout the talks that accuracy of development is mind-blowing. All the axons go to the same place exactly. Some people claim there is some chronotopy, but the next station, all the rules are changing completely. Complete distribution of the axons to the other side. Okay. The next station, like I told you, is the amygdala, okay, which you also saw in the Richard Axel paper here, condensed axons in the amygdala. And he had a paper this year in Nature, again Axel, that he specifically disinhibits these axons and claims that they are important for these innate behaviors of the animal. So again, if you recall, we look back, very similar to what I showed you in Drosophila and C. elegans, these on and off, very strong and clear behaviors. What he's doing here is he puts the mice in this four-room, in a big chamber, and tracks the animal. So this is a behavioral plot, showing how much time the animal spends in each quadrant of this room. Okay? And what you can see, and when he puts no odor, the mouse runs around and spends roughly an equal time in all four quadrants. And then he puts this odorant here in this quadrant of the room. This is a TMT, which is this. You saw it before. It's a component of fox urine, and you see that the mice avoid this quadrant of the room. So this would be this graph, the maximum avoidance. He tests all these odorants. He finds that 2-phenylethanol is an attractive odorant for the mice because they spend more time, and this would be here on this graph. Again, we saw this in the Sakano papers. It has peanut butter. This is also peanut oil. They like peanut butter. But these odorants have no effect. Okay? They don't care about odorant aldehydes and some water molecules, etc. What they did then is that they put their fiber optic of halorodopsin, so they labeled all the mitral cells with halorodopsin, and put their fiber in the medial amygdala. And what they can do is play God, okay, and switch on and off the behaviors of these innate behaviors of the mice. So if mice were disgusted or afraid, avoided to go to the TMT, if they shine light on the specific fibers of the mitral cells that go to the medial amygdala, they can change this behavior completely. Okay? So if normally this mouse would be inhibited or avoid the TMT, when they shine light, the complete avoidance is gone, and they no longer are afraid from the TMT. They also show it for the attracting odorant, and they claim that this is the circuit for innate attraction and avoidance, but this is the innate part. Okay, you remember that the learned part is in the other, not in the medial amygdala. Okay, and they also show this by activating it. So this is really playing God. Okay, taking mouse, putting channel rhodopsin in the neurons of the relevant region in the amygdala. I won't exhaust you. I don't have time. I don't have time to tell you. This is a very nice trick of how they can trap the neurons using genetic tricks and label only the neurons that were active when the TMT was on. So basically, they take a mouse, they put an odor, a TMT odor on, and only the cells that were active when the TMT was on will now express channel rhodopsin. And then they take the mouse, put them in the room, goes around, there's no odor, they turn on the light, and you will avoid going to this side of the room. Okay? What? Yeah, so this is a Cree-R based on immediate early gene. This is ARC in this case. Yeah. So Karen in our laboratory is doing exactly this, trying to repeat this experiment. Okay, the other relay station is to the entorhinal cortex, which feeds the hippocampus. In fact, when you record from hippocampus, you get responses from odor stimuli. So all I want to tell you is that what we learned about the olfactory system is a lot about the olfactory receptor neurons, some about the olfactory bulb, and now it projects out to different stations, and it gets more interesting, more complicated, etc., etc. And last but not least, we have 1 minute, again, just to promote one of our papers that Lior Cohen in our lab, looking in auditory cortex. And what we found is that when the mothers, we studied plasticity of the mothers and the babies, and her babies, when the mother smells her own pups, it causes dramatic changes in the auditory cortex. So when the mother mouse, if you record